Hello, friends. I'm again here with you to discuss a very short and a brief point which one of our friends has raised. The problem was how to distinguish between the different scenarios where B9 and B12 are getting involved. Okay, so there can be some points of confusion in this, but if you know the basics and the important points for each of them, then it can be a straightforward question. So let us quickly look at B9 and B12. First and foremost, what you have to understand, B9 and B12 are interlinked. Okay, B9 and B12 are interlinked. In what manner? The B12 is involved in the recycling of B9. This is first point to keep in mind, that B12 is involved in the recycling of B9, meaning if there is a deficiency of the B12, then the recycling of B9 will not take place. What this means, when there is deficiency of B9, we will have a set of symptoms, right? Now, if B12 deficiency is there, then these signs and symptoms will also be there, along with the unique signs and symptoms which are due to B12 deficiency. If you understand this, then our work is very, very simple. I'll repeat. B9 deficiency will cause some signs and symptoms. B9 recycling requires B12. So if there is B12 deficiency, there will be B9 deficiency, because B9 is getting trapped and it cannot be recycled. So there will be B9 deficiency. So whatever signs and symptoms are there in B9 deficiency will also be there in B12 deficiency. In addition to that, B12 deficiency will have some more signs and symptoms, meaning B12 deficiency will give us a bigger cluster of signs and symptoms. Lesser signs and symptoms will be there in the B9 deficiency. So if you understand this, our work again is simplified. There are 4 points to keep in mind which will prop up again and again and again. Number 1 is megaloblastic anemia. Right, the megaloblastic anemia. Why megaloblastic anemia? Because B9 is required for the purine synthesis. It is required for the conversion of the uracil to thymine. So the purine, pyrimidine both require the B9, and therefore when the B9 deficiency is there, the formation of DNA, RNA doesn't take place. The cell attempts to mature, but it is not able to mature. Particularly the RBCs, they become large. So we get the megaloblastic anemia. Second, you are going to see the incidence of homocystinemia or homocystinuria. I'll show the set of reactions which are involved in this phenomena, and then the picture will become more clear. So from our prior discussion, what you have to remember, they will be present in both means B9 and the B12 deficiency. Now the B12 deficiency will give you some additional features. What are the additional features? You will have subacute combined degeneration of the spinal cord, right? Commonly written as SACD or SCD. In the question, this will be there, SACD or SCD. Please keep in mind that SCD plus the megaloblastic anemia, this will be known as what? Anyone? This will be known as the pernicious anemia. So in the question, the pernicious anemia term can also be there. And the second thing that you will see is the methyl malonic aciduria. Second thing that you'll see is methyl malonic aciduria. These two will be exclusively seen only in B12 deficiency. So clinically now our picture becomes clear. So only megaloblastic anemia plus homocystinemia or homocystinuria, then it is most probably B9. But along with that, we also see the neurological features. and the methylmalonic aciduria is also there, then it becomes a B12 deficiency. If the question says that homocystinemia is there but methylmalonic acid levels are normal, then B12 deficiency is not there, it is the B9 deficiency. Megaloblastic anemia features are there. The picture is given from the peripheral smear, but you are not finding the neurological features. Then we are looking at the B9 deficiency, not the B12 deficiency. Okay, now let us look at the other important points which you have to keep in mind. This is how the clinical picture can be distinguished between B9 and B12. What are the other points that you have to remember? Okay, let us talk about the B9 first. Okay, for B9, you know what is the important point, that B9 will be present as tetrahydrofolate and it is the methyl group donor. So different forms of the tetrahydrofolate can be there. You can have the formyl tetrahydrofolate, you can have the methinyl tetrahydrofolate, you can have the methylene tetrahydrofolate, you can have the form imino tetrahydrofolate. So different types of tetrahydrofolate will be there which are participating in a lot of reactions. So in this context you have to remember three very important lines. They can come as straight line questions. Number one, the most important donor for the methyl group in the different forms will be serine. Serine in the process gets converted to glycine. I'll show you a reaction where we'll see this is happening. Then the important point where the methyl groups are added to the tetrahydrofolate results in the formation of the methylene tetrahydrofolate. It results in the formation of methylene tetrahydrofolate. In the tissues, the tetrahydrofolate is commonly present as methyl tetrahydrofolate. This is where the B12 comes into play. It converts the methyl tetrahydrofolate first to tetrahydrofolate, and then this tetrahydrofolate can get activated into the different forms of the tetrahydrofolate. Please note that in to identify the deficiency, we measure the level of Figlu, particularly in the urine. This is done after providing a loading dose of histidine, right? We provide a loading dose of histidine and then we measure the Figlu in the urine. In the process of the metabolism of histidine, one of the intermediates is Figlu, right? And this Figlu is then converted into the glutamate. For this purpose the tetrahydrofolate is required. Tetrahydrofolate gets converted into the form imino tetrahydrofolate and the glutamate will be formed. If the tetrahydrofolate is not there, obviously this reaction will get blocked and Figlu will accumulate, will pass out in the urine. Another important point we have to remember that the deficiency during pregnancy in the first trimester or before conception will predispose to the neural tube defect. The common term that we use here is the neural tube defect. Okay, that is why supplementation with the folate before conception and in the first trimester is insisted upon. It is seen that it drastically decreases the incidence of the neural tube defect during the delivery. Okay, in addition to this, please look at how the tetrahydrofolate metabolism can be targeted in the different types of treatment. If you target the human enzyme using the methotrexate, then we can make use of this. We can make use of this in the cancer chemotherapy. If you target the bacterial tetrahydrofolate, in that case you get the antibacterial drug, and if you target the enzyme in the protozoa, in that case you get the anti-malarial drug. Okay, so same metabolism targeting the different enzymes in the different types of organism, you can have the different clinical applications. Okay, please keep this point in mind. All of you know that the B12 deficiency we just discussed will cause the functional folate deficiency. We call this as the folate trap. Okay, we'll call this the folate trap. Important point to remember here is the folate trap is there because recycling is not taking place, meaning if you provide some extra folate from outside, this trap might be bypassed, but this extra folate will start pulling some of the available B12 for its recycling. So if you are at a stage where the neurological deficit is not there or it is less, then the neurological deficit can either become manifest or it can become exaggerated. So B9 supplementation in the background of B12 deficiency will exacerbate the problem of the B12 deficiency, whereby the neurological deficit will become much more pronounced. The neurological deficit will become much more pronounced. We already mentioned that the folate deficiency will cause the megaloblastic anemia, right? And the folate supplements have a small problem. Excess folate supplementation may increase the risk of developing colorectal cancer among people with the pre-neoplastic colorectal polyps. Okay, please keep this in point in mind, that folate supplementation has been found to increase the risk of developing colorectal cancer among people with the pre-neoplastic colorectal polyps. So these are the additional points about folate which you have to remember. All of you know this, that causes the megaloblastic anemia. All of you know this, that it unmasks the B12 deficiency. You know the incidence of folate trap. You know the incidence of neural tube defect, FIGLU as the marker for the intracellular deficiency, and the various points related to the addition of methyl group to the tetrahydrofolate. That is about the B9. Let us quickly take a look at the B12 also. In case of B12, what you have to remember, first point, that it is found only in foods of animal origin. It is found in foods of only animal origin. In our body, two well-known enzymes which will require the B12, methyl malonyl coenzyme mutase and the methionine synthase. We'll come back to this methionine synthase in a short while. Coming back to methylmalonyl coenzyme A mutase, from where do we get this methylmalonyl coenzyme A? Methylmalonyl coenzyme is formed from multiple sources. As you can see, it is an intermediate in the catabolism of valine. It can be obtained by the carboxylation of propionyl coenzyme A. This propionyl coenzyme A is coming from the various different metabolic pathways. It can come from the metabolism of the isoleucine, cholesterol, and the odd chain fatty acid. This is very commonly asked. The origin of propionyl coenzyme A from odd chain fatty acid. It can also be obtained directly from the propionate. Okay, directly it can be obtained from propionate. Now, first coming to how the B12 absorption is taking place. So here, very important 4 factors which you have to keep in mind. What are the important 4 factors in B12 absorption? B12 absorption requires two binding protein. They are secreted from different places in our body. The cobalamin is released from the oral mucosa. So if there is any damage to the oral mucosa, or if the secretions of the oral mucosa are decreased, then the cobalamin production will also be decreased. For example, if you have what is called as the dry mouth syndrome, the keratoconjunctivitis sicca, in that case the production of the cobalamin will obviously be less. The intrinsic factor, on the other hand, is coming from the gastric mucosa, right? It is coming from the gastric mucosa. So if there is any damage to the gastric mucosa, then the production of this intrinsic factor will be decreased. This can be seen in conditions like the atrophic gastritis, right? And lastly, what you have to remember is the absorption occurs exclusively in the ileum. The other parts of the small intestine are not able to absorb the B12. Absorption occurs only in the ileum. So what you have to keep in mind, the oral mucosa has to be intact. The oral mucosa has to be intact, number 1. The gastric mucosa has to be intact, number 2. the ileum has to be intact, number 2. Anything else which is required for the successful absorption of B12? See, a lot of production occurs in the gut bacteria, but the absorption is not successful. Why? Because absorption has to occur only in the ileum. Gut bacteria is there mainly in the large intestine. Backflow of the B12, the cobalamin will not occur into the small intestine. Okay, so absorption in the humans will be comparatively less. In lower animals, the absorption can be significant from the gut flora. Okay, so what you have to remember, when we consume the food in the mouth, the cobalamin will bind with the B12 and it will protect the B12 from degradation in the stomach from the acid. Now, when you reach the small intestine, this combination that you have, that cobalamin attached to the B12, this is acted upon by the pancreatic enzymes. This is acted upon by the pancreatic enzymes. What this means is, if there is a problem in the pancreatic secretion, okay, meaning if there is biliary obstruction, in that case the B12 will not get released from cobalamin, and therefore it will not get attached to the intrinsic factor. So the fourth thing which comes into the picture is the pancreatic enzyme. So if there is any problem in the biliary tract, if the pancreatic secretions are not reaching the small intestine, that is the second part of duodenum, then the B12 will not be released, it will not get attached to intrinsic factor, and therefore the final absorption will fail. The intrinsic factor along with B12 are together absorbed. So if intrinsic factor is not able to bind to B12, B12 absorption in the ileum will not take place. So remember, not only the oral mucosa, gastric mucosa and ileum, but we also need adequate pancreatic enzyme secretion for successful absorption of the B12. I already told you that we have what is known as the pernicious anemia. Pernicious anemia will include two things. One is the megaloblastic anemia, and second is the irreversible degeneration of the spinal cord, commonly known as SCD or the SACD, subacute combined degeneration of the cord. Now let's look at a little bit of chemistry. Okay, so the neurological deficit is said to be due to defect in methylation of an arginine residue in defect in methylation of one arginine residue in myelin basic protein. Myelin basic protein is a protein which causes the myelination of the axon. It causes the deposition of the lipids in the axon. So if this myelin basic protein doesn't become active, then the myelination will not take place. It will cause the degeneration of the neurons. Looking at where the methylmalonic coenzyme is produced in the deficiency of the B12. See, I told you that propionyl coenzyme is the source for methylmalonyl coenzyme. Propionyl coenzyme coming from the odd chain fatty acid. Remember, I told you it can come from the odd chain fatty acid. So metabolism of propionyl coenzyme into the methylmalonyl coenzyme, and then the methylmalonyl coenzyme mutase. Remember, I told you it is one of the two enzymes which importantly requires the B12. So if B12 deficiency is there, conversion of methylmalonyl coenzyme to succinyl coenzyme will not take place. Okay, and the methylmalonyl coenzyme will accumulate. It will come out in the urine, resulting in methylmalonic aciduria. It will result in methylmalonic aciduria, okay, which is one of the important findings in the B12 deficiency. Okay, so the neurological deficit and the methylmalonic aciduria characteristic of the B12 deficiency. Let's look at now the homocystinuria and the folate trap. So what happens, that this methyl tetrahydrofolate I told you is converted into the tetrahydrofolate. Okay, the enzyme which is acting here is the methionine synthase, requires the B12. It requires the B12. The methyl tetrahydrofolate doesn't have any other action. It is primarily used in this particular reaction, meaning if the B12 is not there, then methyl tetrahydrofolate cannot be converted to tetrahydrofolate, and further activation of tetrahydrofolate will not occur. This will result in the folate trap. This will result in the folate trap. Okay, so folate trap is clear. Folate trap is clear that if B12 is not there, then the tetrahydrofolate gets trapped as the methyl tetrahydrofolate. Now this methionine synthase converts homocysteine. It converts the homocysteine into the methionine, meaning if the tetrahydrofolate is not there, then homocysteine will accumulate, resulting in homocysteinemia and the homocystinuria. Okay, and from where this homocysteine is coming, the methionine, it is getting converted into the SAM. It gets converted into the SAM. The SAM will the SAM will provide the methyl group to the glycine, resulting in the formation of sarcosine, gets converted into the S-adenosyl homocysteine. S-adenosyl homocysteine is acted upon by the hydrolase, whereby adenosine and the homocysteine are released. So the methionine is getting converted to homocysteine. Recycling requires the B12 and the folic acid, meaning if B12 is not there, folic acid will get trapped, the conversion will not take place. So this homocystinuria we will see in both the B9 and the B12 deficiency. Okay, so these are the very pertinent factors that you have to keep in mind, and I just told you in the beginning itself common features that you will see in both B9 and B12 deficiency, that is the megaloblastic anemia and homocystinemia or homocystinuria. The features that you will see only in case of B12 deficiency, that is the neurological deficits, the subacute combined degeneration of the cord, the SCD or the SSCD, along with the methyl malonic aciduria. Along with the methyl malonic aciduria, these will be the features exclusive to the B12 deficiency. So I hope with this discussion you are quickly able to identify the important features about B9 and B12. You know the questions are being asked again and again and again. For more content like this, or if you have any doubts or queries how to find out the differences or the concept based questions if you have from biochemistry, you can drop a message on any of these platforms. We are available on the Telegram, we are available on Facebook, on YouTube, on WhatsApp, on Instagram. The keywords which you can use is biochemistry by Dr. Nilesh Chandra. Thank you very much. Stay tuned, keep watching. Bye bye.